Hello everyone, today I'm excited to share our research plan poster on the effects of transcranial direct current stimulation, or TDCS, on symptoms of chronic cognitive fatigue. So this study will be conducted by our team at the University of Central Lancashire in collaboration with Plita Science from Copenhagen. The motivation behind this research stems from the increasing interest in non-invasive brain stimulation techniques, such as transcranial direct current stimulation, or TDCS. So this technique has emerged as a promising tool due to its ease of use, effectiveness, and minimal side effects profile, especially when compared with current medical treatments. So for those unfamiliar with TDCS, this is a neuromodulation technique that delivers a constant low electrical current on the scalp via electrodes. Usually there is an anodal electrode and a cathodal electrode, but this is not always the rule. So this current modulates neuronal activity, potentially enhancing cognitive functions, and this technique is celebrated for being non-invasive and user-friendly, making it accessible for various applications, including clinical and everyday settings. So we will aim to explore whether TDCS can effectively reduce symptoms of chronic fatigue, which is particularly relevant in today's context of increased mental strain due to the COVID-19 pandemic and long COVID symptoms. So in terms of methods, we aim to recruit 150 fatigue participants, and these fatigue levels for inclusion will be measured via the Modified Fatigue Impact Scale questionnaire. And participants will be divided into two groups, an active TDCS group and a sham placebo group. So the electrodes placement will follow the standard 10-20 EG system to target the dorsolateral prefrontal cortex, or the LPFC, with a nodal electrode on F3. Participants will use a portable TDCS device at home for 60 days, and during this time, a subgroup of participants will be provided with Empatica 4 bands to measure physiological data such as heart rate, and this subgroup of participants will also be asked to perform an EEG short computerized task at the lab and complete a modified fatigue impact scale pre and post intervention, and at a 3-month follow-up to observe if TDCS application is effective in reducing symptoms of fatigue and if these effects are long-lasting. In terms of analysis, our analysis will focus on comparing the cognitive performance on the short EEG lab task and fatigue levels between the active and sham groups, and also, more importantly, within samples pre and post intervention and at the 3-month follow-up. We will employ statistical methods to determine the significance of differences observed, and in particular, we will analyze correlation and regression to observe the relationship between TDCS intervention and fatigue scores provided by the participants based on individual differences. So in conclusion, we are looking forward to start to collect our data and gain more insights into the effects of TDCS and symptoms of chronic fatigue, as well as investigating potential electrophysiological markers of this condition via our EEG observations. Thank you for your attention, and I look forward to any questions you might have.